Imagine a world where we could track down criminals from a single drop of blood. Imagine identifying ancient diseases from the bones of dinosaurs. Imagine diagnosing deadly infections with just a swab of saliva. This isn't science fiction, this is the power of PCR. PCR stands for Polymerase Chain Reaction. It's a technique that changed the world of biology and medicine forever. With PCR, scientists can take a tiny amount of DNA and amplify it billions of times over. This breakthrough opened up a universe of possibilities. Suddenly we could study DNA from almost any source. We could analyze genetic material from crime scenes, ancient remains, and even single cells. The implications were staggering. PCR, in essence, gave us a powerful new lens to examine the very building blocks of life. It allowed us to explore the intricate code that makes us who we are. And like any revolutionary tool, its impact continues to ripple through countless fields of science and technology today. Before we delve into the magic of PCR, let's take a moment to appreciate the molecule at its heart. DNA. DNA is the blueprint of life. It's a long, winding molecule found in the nucleus of almost every cell in our bodies. Imagine DNA as a vast library. This library contains all the instructions needed to build and maintain a living organism. These instructions are encoded in the sequence of 4 chemical bases: adenine, A, guanine, G, cytosine, C, and thymine, T. These bases pair up in a specific way, A with T and C with G. This pairing forms the rungs of the famous DNA double helix, resembling a twisted ladder. The order of these base pairs is what determines our genetic code. It's what makes us unique. PCR allows us to zoom in on specific sections of this vast DNA library. It's like having a magic key to unlock and copy any book we desire, no matter how small or hidden it may be. PCR is elegantly simple in its concept. It mimics the natural process of DNA replication that happens in our cells all the time. But instead of replicating the entire DNA molecule, PCR focuses on a specific region of interest. Think of it like photocopying a single page from a massive encyclopedia. You don't need to copy the whole encyclopedia, just the information you're looking for. The PCR process involves a repeated cycle of 3 key steps. 1. Denaturation. The DNA double helix is unzipped by heating, separating the 2 strands. 2. Annealing short DNA segments called primers attached to specific matching sequences on the separated strands. These primers mark the boundaries of the region we want to amplify. And 3. Extension. A special enzyme called DNA polymerase builds new DNA strands, starting from the primers, using the original DNA strand as a template. Each cycle essentially doubles the amount of the targeted DNA region. Repeat these cycles over and over, and you can generate millions or even billions of copies from a minuscule starting amount. Imagine the DNA double helix as a zipper. The teeth of the zipper are the base pairs held together by hydrogen bonds. In denaturation, we apply heat, much like running a hot iron over the zipper. This heat breaks the hydrogen bonds, causing the 2 DNA strands to separate. Now we have 2 single strands, each ready to serve as a template for creating new DNA. This separation is crucial because DNA polymerase, the enzyme responsible for building new DNA strands, can only work on single-stranded DNA. The temperature required for denaturation depends on the DNA sequence. Longer sequences with more G and C base pairs, which have 3 hydrogen bonds, require higher temperatures to separate. Section 5. Finding the target, annealing. Now that our DNA strands are separated, imagine we have 2 single-stranded templates floating around in our reaction tube. Our goal is to amplify a specific region on these templates. This is where primers come in. Primers are short synthetic DNA sequences designed to match the flanking regions of our target DNA. They act like bookends, marking the start and end points of the region we want to copy. During annealing, we lower the temperature, allowing these primers to bind, or anneal, to their complementary sequences on the separated DNA strands. The specificity of primer binding ensures that only the desired region of DNA is amplified. Section 6. Building the copies, extension. With the primers in place, it's time for the star of the show, DNA polymerase. This remarkable enzyme is responsible for building new DNA strands by adding nucleotides, the building blocks of DNA, one by one. DNA polymerase needs a few things to get started. It requires a single-stranded DNA template to copy, a primer to provide a starting point, and a supply of free nucleotides A, T, C, and G to build the new strand. During the extension step, the reaction temperature is raised to the optimal temperature for DNA polymerase activity. The enzyme starts at the primer and moves along the template strand, adding complementary nucleotides to build a new DNA strand. For example, if the template strand has an A, DNA polymerase will add a T to the new strand. If the template has a C, it will add a G, and so on. This process continues until the entire target region has been copied. Section 7. The power of exponential growth. At the end of one PCR cycle, we've doubled our starting DNA. We now have 2 copies of our target region. But the real magic of PCR lies in its repetitive nature. Each cycle doubles the amount of DNA. This means that after 2 cycles we have 4 copies, after 3 cycles 8 copies, and so on. This exponential growth is the key to PCR's incredible sensitivity. After 20 cycles we have over 1 million copies. After 30 cycles, over 1 billion. This means we can start with a minuscule amount of DNA, even a single molecule, and amplify it to a level where it can be easily detected and analyzed. This ability to amplify DNA from tiny samples has revolutionized fields like forensic science, where evidence is often limited, and ancient DNA research, where samples are often degraded and fragmented. Section 8. PCR in Action. Medical Diagnostics. Perhaps the most impactful application of PCR is in medical diagnostics. PCR can identify the presence of viruses and bacteria in our bodies, even at very low levels. Imagine you have a sore throat. A simple swab of your throat can be analyzed using PCR to detect the presence of the virus or bacteria causing your infection. This rapid and accurate diagnosis allows for timely treatment and prevents the spread of infectious diseases. PCR is also used to diagnose genetic disorders. By amplifying specific regions of a patient's DNA, doctors can identify mutations or variations associated with inherited diseases. This information helps in early diagnosis, genetic counseling, and developing personalized treatment plans. Section 9. PCR's role in forensic science. In the world of forensic science, PCR is a game changer. It allows investigators to analyze DNA evidence from crime scenes with incredible precision, even from tiny samples like a single hair or a drop of blood. By comparing DNA profiles from crime scene evidence to those of suspects or victims, forensic scientists can link individuals to a crime scene or exonerate the wrongly accused. PCR has become an indispensable tool for solving crimes, bringing justice to victims, and ensuring accurate convictions. PCR is also invaluable in paternity testing. By comparing specific DNA markers between a child and the alleged father, parentage can be established with a high degree of certainty. Section 10. PCR and the future of scientific research. PCR's impact extends far beyond medical and forensic applications. It has become an essential tool in countless research labs worldwide, driving discoveries in fields like genetics, evolutionary biology, and environmental science. PCR allows scientists to study the genetic makeup of organisms, from bacteria to humans. By comparing DNA sequences, they can trace evolutionary relationships, understand how organisms adapt to their environment, and even design new organisms with beneficial traits. In environmental science, PCR helps monitor biodiversity, track the spread of invasive species, and assess the impact of pollution on ecosystems. By analyzing DNA from environmental samples like soil or water, scientists can identify the presence and abundance of different species, even those that are difficult to detect by traditional methods. Section 11. Conclusion. A legacy of breakthroughs. Since its invention, PCR has revolutionized the way we study and manipulate DNA. It has become an indispensable tool in medicine, forensics, and research, with applications spanning from disease diagnosis to crime scene investigation to understanding the very origins of life. The future of PCR is bright. Scientists are constantly developing new variations and applications for this versatile technique. We can expect to see PCR play an even greater role in personalized medicine, gene editing, and our understanding of the complex web of life on Earth. Like the invention of the microscope, which opened our eyes to the invisible world of microorganisms, PCR has provided us with an unprecedented view into the microscopic world of DNA. It has given us the power to read, analyze, and even manipulate the code of life itself. As we continue to explore the vast potential of this revolutionary technology, one thing is certain: the future of PCR is filled with exciting possibilities.